So welcome back from the break. Let's take a look at these two concept check questions, and then we'll look at the last section, which is all about the role of fungi in ecosystems and our own interactions with fungi. So what features of chytrids suggest that they are members of the basal fungal lineages? The answer is really simple. The flagellated spores. Remember that spores are present, flagella are present on the, or thought to have been present on the common ancestor that is shared by fungi and animals. We certainly see that as a feature of opisthocons in general. And so it stands to reason that if there is a basal member of the fungal lineages that bears, or if there is a clade of fungi that bears flagella, they should be placed basally to fungal lineages, particularly seeing as how the other major lineages, the mucoromycetes, the ascomycetes and basidiomycetes, all lack a flagellated stage. So the presence of fungi or the presence of flagella, get a little tongue-tied, sorry about that. The presence of flagella identifies the chytrids as a basal fungal lineage. How does form follow function in mucoromycetes, ascomycetes, and basidiomycetes? Well, basically here it's things that you can identify. So the mucoromycetes and ascomycetes and basidiomycetes also have spores covered with environmentally resistant coatings to prevent them from dehydrating. That protects them when they're released into the environment. The sporangia have thick walls. In the case of mucoromycetes, the zygosporangia, where meiosis occurs to give rise to the haploid cells that will eventually become the spores, that has a coating, a thick wall hydrophobic coating around it to prevent it from dehydrating. So there's form and function. In the ascomycetes, you have the conidia forming on the tips of the conidiophore, which puts them in an elevated position, which makes it very easy for them to be distributed once they're released. The asci form inside the ascocarps, the fruiting bodies, which protects them from dehydration and protects them from I guess, loss or damage until they are ready for release. So it provides a protective function. In the case of the basidiomycetes, the gill structure on the inside of the mushroom cap, that highly folded structure provides a large surface area for the production of a lot of basidia, which leads to a lot of basidia spores, which means you have a lot more spores to release to increase the likelihood of successful reproduction. So there are some examples. I mean, basically, you can look at the life cycles and take a look at various stages within the life cycle and see structures that are adapted to the function. And if you want the big one, remember that all fungi are absorptive heterotrophs. So when you look at the mycelium and the structure of the mycelium from hyphae, which emphasize a high surface area to volume ratio, there is a structure that is optimized for absorption. So there is a number of features that you could mention here that demonstrate form and function connectivity. Okay, so let's take a look at the roles of fungi in ecosystems and our interaction with fungi, because there are a number of interesting things that go on here. So we do need to remember that fungi are important decomposers. They decompose organic matter and return chemicals back into the environment and make those materials available to both other fungi and also to other types of organisms, most notably plants. So here is a classic structure that is formed by basidiomycetes. This is referred to as a fairy ring because in the Middle Ages in Europe, this was thought to be the product of fairies basically having a dance party overnight. Remember, the mushrooms emerged literally overnight. And the traditional thought was that when fairies got together in the middle of the night and danced around in a circle, wherever a fairy's foot touched the ground, a mushroom would emerge. So in the morning, when they saw this circle of mushrooms, they figured that the fairies had been out dancing again, and this had produced this quote unquote fairy ring. We now know that this is actually the fruiting bodies that are emerging at the tips of the emerging dikaryotic mycelium that is radiating out in a circular fashion from the point of origin of the fungus. And so on the margins of the fungus here are where the mushrooms are being produced. Now, if you look at this ring, you'll also notice that the grass on the margins of the ring looks a deeper green color than the grass on the inside of the ring or on the outside of the ring. And that's because the grass on the margin of the ring is taking advantage of the mushroom's decomposition of organic material in the soil as it's supporting itself, and therefore taking up some of that organic matter that's been recycled back into the soil. And it is evident by the darker green color in the grass that it's benefiting from the fungi's breaking down of the organic matter in the soil. So here you can see the fungi as decomposers. Fungi also form very important mutualistic relationships. We've talked about the plant relationships already. We'll touch on those again, but we also need to talk about mutualistic relationships between fungi and algae and cyanobacteria in the form of lichens, and also symbiotic relationships that fungi can form with animals. They're not just parasites of animals, although that's a big part of it as well. So in the case of plant fungal symbiosis, we've already talked about the mycorrhizae that are incredibly important to the success of plants in general, not only as colonizers of dry land, but also in terms of the success of plants in ecosystems and in agriculture. If you transplant a plant from its local habitat into a new region and its symbiotic fungi is not present, that plant may fail to thrive. It may die, it may grow, but it may grow very poorly. It grows much better in the presence of its fungal symbiont. In the case of some fungal symbionts, they even help protect the plants from herbivores by making chemicals that are unpleasant. So the fungi are actually protecting their plant host from being eaten. And I've already talked about the wood wide web. So let me put this in here. So one of the interesting things about this web structure is that it resembles it's not right. So remarkably, this wood wide web formed by fungi and the trees that they are symbiotic with actually was a forerunner of what we now refer to as the World Wide Web. There is a remarkable similarity between the structure of the World Wide Web and the Wood Wide Web, including hub trees that are central and act as distribution points for signaling between trees attached to the hub. And excuse me, these hub trees actually act as a distribution point for signals coming in from adjacent trees to be distributed out to other trees attached to the network. It's quite remarkable what this looks like. And this is something that we've only just started to really get a handle on. It's something that we've only known about for about 15 years now. But it predates by many millions of years the World Wide Web that we know of as part of our information technology. Kind of remarkable that this should be the case. So here is just a couple of graphs that indicate the advantage of having a fungus present within a plant to protect it from a pathogen versus not having the fungus present to protect it from the pathogen. So where the fungus is present, the plants thrive better in terms of the amount of damage that's done to them. Whereas if the fungus is absent, the leaves have more damage. Again, just a reflection of the fact that the fungus is serving as a protective agent for the plant by producing something that the herbivores prefer not to eat. In the case of animal symbiosis, the most remarkable symbiosis is between ants and plants in terms of what is known as the leafcutter ants. But of course, we've already talked about the fact that many animals, like cows, what are called ungulants, those are the animals that rechew their food. They regurgitate it and chew it a second time before they swallow it. In the second chamber of their stomach, they have not only prokaryotes, but they also have fungi that help break down the plant material because they can break down cellulose. But in the case of the ants, the leafcutter ants, the ants specialize in particular types of leaves that they want to harvest because they also specialize in the types of fungi that they will grow. And it turns out that they practice a form of agriculture that we are all familiar with, what is referred to as a monoculture, which refers, and it's pretty much an industrial monoculture. It refers to the fact that humans, when they practice industrial monoculture, set aside large blocks of land to grow a single crop on. And we spend time and energy and resources to protect that single crop. We fertilize it, we weed it, we spray herbicides, we spray insecticides, we do all sorts of things to try and protect that crop plant from the things that would damage it, so that we can reap the benefit of a large-scale monoculture. Well, the ants have been doing the same thing, only they've been doing it for millions of years. The leaves that they take in, they mulch up. They chew them up and mulch them up, and then they seed this mulch with spores of the fungus that they want to grow. And they will very determinedly go through and weed out the spores of any fungus that they don't want to grow. They will apply chemicals to kill off anything that they don't want to grow there. And they harvest specialized fruiting bodies that grow on the tips of the fungi that they're farming, because that's what they're going to eat. So they are using the fungi as a way of tapping into the resources of the plant material that they're carting back to their colony by mulching it up and letting the fungi then process that mulched material and producing something that the ants can then eat. So they're farmers in every sense of the word, and they practice a farming strategy that we are very, very familiar with, growing a monoculture over large beds of ground with weeding, with chemical application to prevent the growth of any foreign crop that we don't want, or that they don't want in the case of the fungi. It's a very remarkable system that the ants have been using for, as I said, millions of years. We've only been doing it for about the last 150 years or so with our agricultural revolution. So we're newcomers to the scene when it comes to industrial monoculture agriculture. Then in terms of fungal interactions with cyanobacteria and algae, we see these in the form of lichens. So lichen taxonomists usually assign binomial names to different types of lichens, but a lichen is not actually a single organism. It's a symbiosis between a fungi, typically an ascomycete, and either a cyanobacterium or an algae that is mutually beneficial to both, or at least it's viewed as being mutually beneficial typically. The lichens come in 3 different forms, and you've seen those 3 different forms in the fungal lab. So you have the foliose, which is leafy in appearance. You have the crustose, which forms a crust on the surface that it's growing on. And then the third type is the one that neither looks leafy nor crusty, and that's fruticose. It's typically described as being shrubby, although it may not necessarily look very shrubby in appearance. So here you can see the 3 different types. Foliose is pretty clear cut, very leaf-like. Crustose is very clear cut because it forms this distinctive crust on whatever it is that's growing on. Here it's growing on a rock, but it could be on a tree. And then you have foliose, which is shrub-like, I guess, but may not necessarily look very shrubby in appearance. So it is really a symbiosis between the hyphae of the fungus and the either algal cells or cyanobacterial cells that occupy the mycelial mass and give it its characteristic greenish color as a consequence of the chlorophyll pigments in the cyanobacteria. Remember also that cyanobacteria also form specialized cells that do nitrogen fixation. So the cyanobacteria also are a source of fixed nitrogen for the fungi, which is an added benefit for the fungi, something that they don't necessarily get access to, particularly seeing as how lichen are also considered to be early colonizers of virgin ground, because lichens also have the ability via their fungi component to split rock up into smaller fragments. And that rock splitting is what begins the formation of soil that allows other organisms then to come in, primarily plants, to colonize this ground, but it requires something to split the rock up first into smaller fragments that develops into soil. And lichens are particularly good at doing this via the fungal component of the lichen. So this is what a typical lichen will look like. You can see the hyphae that make up the mycelium of the fungus. Embedded within the mycelium are either the algal cells or the cyanobacterial cells. Typically, the fungi and the algal cells reproduce separately from each other using separate reproductive strategies appropriate for each of them. In this case, we're talking about an ascomycete, so it's going to form an ascocarp. The fungal cells will typically, the algal cells will typically reproduce by whatever strategy is appropriate from them. These are unicellular algae in this case, so they're going to follow a reproductive strategy similar to the one that we saw with unicellular algae when we covered the protists. But occasionally, small fragments of hyphae with accompanying algal cells or cyanobacterial cells will break free of the lichen mass, We refer to these as Soredia, that's the plural, Soredium is the singular. These can then give rise to a new lichen mass because the hyphae grow to produce a new mycelium, and the algal cells reproduce right along with it, and that gives rise to a new lichen. The importance of lichens also is that they are very, very important nutritional sources for animals, particularly those that overwinter in locations where there is substantial snow. The lichens remain intact, attached to rocks and trees and tree stumps and the like. And so these animals can dig down through the snow. This would be elk and antelope and the like, can dig down through the snow and feed on the lichen during winter and get a very nutritional meal from the lichen. And the lichen can, of course, reproduce themselves and regrow once the snow disappears and spring returns. But during winter, these are a very important nutritional source when other nutritional sources may have disappeared from the landscape. Fungi are also incredibly important pathogens. Some of the most nastiest medical photographs that you can see are actually fungal infections of humans. They are pretty disgusting and pretty messy to look at. So if your intention is to head to medical school, but you don't like ugly looking pictures, you're unfortunately in for some really ugly looking pictures, because fungal infections do look pretty nasty. So they're also important pathogens of agriculture. As I said, in some situations, up to 80% of agricultural loss is directly attributable to fungal attacks on either the plants or the stored harvest. Either way, it leads to loss of the valuable material that we have grown. Where they are parasites of animals, as I mentioned before, it's referred to as mycosis, whether it be domestic animals, animals in the environment, or ourselves. It's referred to as mycosis. We'll talk about mycosis in a moment, but here are some examples of fungal infections of plant material. So you've got corn smut. I'm informed that these enlarged corn kernels that are the product of the fungal infection are actually considered in some cultures to be a delicacy. They're actually eaten. I mean, this is similar to eating a mushroom. So, you know, I suspect that it would be no different. Here you've got some fungi growing on the underside of maple leaves, leading to necrosis. That's the death of the tissue and ultimately death of the leaf. Here you've got a type of ascomycete that's growing on rye. In this case, it also grows on barley, that gives rise to a structure called an ergot. That's that purple feature that you can see there. Where this is important is because ergots produce some very interesting chemicals. The most interesting one of all is produced by ergot growing on rye, and it's referred to, it's known as lysergic acid. Now, lysergic acid was first identified as a product of the ergot producing fungi on cereal grains in Europe in the 1930s. And the most famous chemist to work with this was a chemist working for a Swiss chemical company, and his name was Albert Hofmann, otherwise known as Abbie Hoffman. And the reason why Abbie Hoffman is important is because he spent some time working with the chemistry of lysergic acid and coming up with some chemical modifications of lysergic acid. And the one that is most famous is one that he identified as lysergic acid DD25. So this is his 25th modification. And its importance is spelled out in the name and the initials that come from the name. So the full name of this modification is lysergic acid diethyl amide. That's where the DD comes from. So lysergic acid diethylamide, abbreviated LSD. That's right. LSD is a chemical derivative of a chemical, lysergic acid, that is produced by ergots growing on cereal grain, specifically rye and barley. And Abbie Hoffman is famous for having the first LSD trip. The first one was accidental. He accidentally absorbed some of the LSD through his fingertips and noticed that he felt really strange, but didn't make the connection to LSD until a few days later. Then he deliberately dosed himself with a small amount, microgram amount of LSD. And then for some reason, which he hasn't fully answered, chose to ride his bicycle home while under the influence of LSD. And this has led to, in some circles, a very important celebration known as Bicycle Day. It's to celebrate the day that Abbie Hoffman had the first deliberate LSD trip and rode his bicycle home under the influence of LSD. This is the origin of the term trip when taking LSD, because Abbie Hoffman took a trip on his bicycle, literally and figuratively, under the influence of LSD. LSD has received a very bad rap over the years because of its hallucinogenic activity. It is classified as a class 1 drug. It has been traditionally referred to as a chemical that has no known medical benefit, and therefore, as a class 1 classified drug, it has been until recently been impossible to do any sort of psychological or medical research involving LSD in the United States. In some other countries, that has been possible. And more recently, it has become possible to do research using LSD. And so in the resources page for this chapter, I've included some references to research on LSD, including the role of the CIA in introducing LSD to the United States, because the CIA also researched the use of LSD as a psychogenic drug that may be useful for influencing the mental state of individuals and therefore being able to influence their behavior. It turned out extremely badly. In one case, one of the unwitting subjects, because the CIA did a lot of this research without letting individuals know that they were being dosed with LSD, stepped out of the window of a 10-story building. And obviously that didn't work out well. And the CIA for many years denied that it was responsible for any of this, but eventually the classified documents were released that clearly demonstrated the role of the CIA in this research. So there's a link for that. And there's also a short video about Bicycle Day and Abbie Hoffman and the discovery of LSD. So you can take a look at that. The links are in the resource page. Then in the case of chytrids, I mentioned that chytrids have been parasites of amphibians, and the way in which it's been demonstrated that it is a fungus is by treating several areas where the die-off of amphibians has been noticed with fungicides, and that has led to the survival of the frogs or the salamanders, depending on what amphibians you're working with. And this effect has been seen around the world. So in a number of different countries, in Australia, in Southeast Asia, here in America, in some locations in Europe, we've seen this die off, and in each case it's been associated with 2 different chytrids that are infecting the amphibians. And this is considered to be a bellwether signal regarding the health of the ecosystem. Because when the amphibians die off as a response to the chytrid infections, it indicates that the ecosystem is not functioning healthfully, that there are some issues going on with the ecosystem. And so once you see this amphibian die off, it's considered to be a warning that the ecosystem is in trouble. But it's the chytrids that are responsible for this. Then in terms of human parasitism, the mycosis that you might be most familiar with is ringworm. The same fungus also forms athlete's foot. It also gives rise to jock itch. It also gives rise to tinea, and it also gives rise to a scalp infection. It's the same fungus. It's just different strains of that fungus taking up residence in different locations and giving rise to different symptoms. There's also a fungal infection called coccidioidiomycosis. I think I pronounced that correctly, which gives tuberculosis type symptoms, and it is lethal if left untreated. And again, people with compromised immune systems are really prone to these sorts of conditions because the immune system can typically deal with the fungal spores and prevent the infection. But with a compromised immune system, that's not always the case. You may also be familiar with Candida infections. Candida is a form of yeast. It is a fungus, and so this is considered to be a yeast infection. And it is an infection of the vagina. These cells are often cohabitants of the vaginal epithelial tissue, but are kept in check by the immune system. But again, when the immune system is compromised, or the pH of the vagina is compromised in some fashion, these organisms, as opportunists, then start to grow. Fortunately, they can be treated. You may also be familiar with a type of yeast called Rhodotorula, otherwise known as red yeast, or you may not be familiar with it. But if you see a red mold-like growth in your shower enclosure or on the shower curtains or something like that, this is actually Rhodotorula. It's not considered to be pathogenic, but it grows in regions that are warm and moist, and your shower enclosure, your shower curtain, your bathroom in general is a pretty good environment for it to grow, because it can metabolize body oils, leftover soap, and things like that as a source of organic material to process. And of course, it's in a moist environment with a warm environment, which most bathrooms are. It can be killed by using dilute bleach, but there's no real way of removing it completely because the spores are there all the time, and it's just a matter of time for those spores to land back in the environment again and start growing. So if you see some of these types of growths, these little red mold-like growths, they're not pathogenic. They don't look pretty, but you can remove them with bleach, but you'll have to keep an eye out for the reoccurrence, because it's almost certain that it's going to reoccur again. Then, if you like blue cheeses, the blue veins that run through the cheese are actually due to fungi that have been injected into the cheese, and those fungi are using proteins and fats in the cheese as a food source, and are growing, and they change the nature of the cheese, and they add a very distinctive flavor to the cheese. They also soften it as a consequence of their growth. Of course, if you enjoy adult beverages, alcoholic beverages, that's a product of fermentation, which is again a fungal feature, yeast in this case, and also in the case of bread and any other baked product that uses yeast as a leavening agent. In both cases, in the production of alcoholic beverages and in the production of bread, the yeast are functioning anaerobically and are therefore carrying out fermentation. The alcohol is retained in alcoholic beverages. You might ask, where's the alcohol in the bread? Well, of course, you bake the bread at high temperature, so the alcohol is baked out of it. But it does and can leave behind distinctive flavors as a consequence of the yeast's activity. And so there are several different varieties of Saccharomyces cerevisiae, which is the yeast that is used in bread baking and producing alcoholic beverages. But in bread baking, there are several strains of yeast that you can use, and they give their own distinctive flavor to the bread, depending on the strain of yeast that you use. Then, of course, there's the penicillium fungus. There are multiple strains of the penicillium fungus that produce penicillin. Some are more industrially relevant than others because of the amount of penicillin that they actually produce. And so the strains that are used pharmaceutically are strains that have been identified during the Second World War as strains that produce significant quantities of penicillin. Of course, penicillin was first discovered back in the 1920s, but its significance was not recognized until the mid to late 1930s, but it was not till World War II that the industrial production of penicillin actually began as an antibiotic to treat bacterial infections. And then Saccharomyces itself is also used as a biotechnology vector in the same way as bacteria are used. The big advantage of putting genes into yeast is that yeasts as eukaryotes can read eukaryotic gene sequences. And just in case you've forgotten why that's significant, you should remember that eukaryotic genes contain both introns and exons. Introns are non-coding regions, exons are coding regions. And in order for the gene to be read correctly, during the production of the messenger RNA from the original transcript, the introns have to be cut out and the exons spliced together to form the functional messenger RNA. If we want to introduce the eukaryotic gene into bacteria for the production of a protein like insulin or blood clotting factor, we have to do that externally, then create a DNA copy of the edited messenger RNA, which we can do, and then insert that double-stranded DNA copy of the now edited gene into the bacteria so that the functional gene is what is read by the bacteria, because bacterial genes do not have introns and exons. Putting the gene into something like a yeast cell means no editing required. The yeast does the editing itself because it has the same system that we have, and it does the excision of the introns and the splicing of the exons all by itself. We don't need to carry it out ahead of time. It's just that it's a little more difficult to insert a gene into yeast, but there is a yeast equivalent to a bacterial plasmid. You may recall that we talked about bacterial plasmids when we did the prokaryotic chapter 27. The yeast equivalent is called a YAC, Y-A-C. That stands for yeast auxiliary chromosome. It is a circular DNA, and you can insert genes into that, and you can then insert the YAC back into the yeast cell and have the gene that you inserted expressed inside the yeast cell, and the yeast can then produce the protein of interest without any further editing other than what the yeast will do for itself. So I'll go back to this slide. So this is just an indication of the ability of penicillium to produce the penicillin antibiotic and its effect on Staphylococcus, which happens to be a bacterium that is sensitive to penicillin. Penicillin is not a universal antibiotic. It does not work against gram positives. It works against gram negatives. And that's because of the nature of the cell wall. If you're not sure about that, go back and take a look at chapter 27, where we discuss gram positive and gram negative. So this is the final slide. This is just a nice summary chart of the diversity of fungi, the phylum names as we have discussed them, and the classic features that identify each of the clades, something that you probably want to use as a starting point for understanding the diversity of fungi. Final concept check before we finish. List some of the benefits algae can derive from their symbiosis with fungi that should be lichens, not leeches. So what are the benefits? They get access to nutrients and water because of the fungi's ability to extract nutrients and water from soil. If they're colonizing virgin ground, the fungi can split rock to begin soil formation, and in the process, again, access minerals and water that the algae themselves probably couldn't. They provide a protective environment for the algae. And in terms of reproduction, when the Soredia break free, they carry the algae with them so that they can enter new environments and reproduce and grow in large numbers in that new environment. So those are some benefits that you could list in terms of algae in their interactions with fungi and lichens. What features of pathogenic fungi permit their efficient transmission to a new host? The feature here is the feature that all fungi possess, and that is environmentally resistant spores and large numbers of spores. So if you produce a spore that can resist drying out, but remains viable for a long period of time in the environment because it resists drying out, and if you can produce significant numbers of spores, that increases the likelihood that at least one spore can infect a new host, then you increase the likelihood of transmission from one host to another. And for pathogenic fungi that have the capacity to kill their host, it's really important that their transmission to a new host be very efficient and have a high likelihood of success, because if there isn't, when the fungi kills its current host, that also means the end of the fungus and very little opportunity for transmission to a new host. So having highly resistant, highly viable spores or long-lived spores that can remain functional and viable in the environment, and with large numbers of spores, literally millions and billions of spores being released, simply increases the likelihood of successful transmission from one host to the other before the death of the original host. So that's fungi. Very, very interesting group of organisms. A lot of interesting things there for you to learn. A lot of more interesting things that you can chase down using some of the links in the resources section if you find that you want to know a little bit more about fungi because you've suddenly discovered that there are some cool things or some interesting things that you didn't know before. So take a look at those resources and see if and learn a little bit more if that's what you want to do. As always, if you are wanting to ask questions or you have concerns or comments about anything that I've covered here, by all means, come and see me during office hours. Otherwise, I'll see you back here again when we finally start talking about animals, by talking about embryonic development and reproduction in animals. I'll see you then.